If nerve impulses are carried along chains of neurons, as we've been saying, how are they transmitted between neurons or between a neuron and its effector organ? This was a question that plagued Otto Loewy in the 1920s. One night, Otto dreamt about an experiment using frogs' hearts that he hoped would answer his question. When he woke up, he rushed to his lab to set up this experiment where he placed two hearts in separate but connected chambers filled with saline. When he stimulated the vagus nerve of one heart, it caused that heart to beat slower, but it also caused the other heart, in which the nerves were denuded, to slow down too. Otto hypothesized that the first heart was able to affect the second by releasing a chemical into the saline that flowed between the two chambers. This discovery of chemical transmission, and specifically of what is now known as acetylcholine, earned Otto the Nobel Prize in 1936, which he shared with Sir Henry Dale. This module will discuss how neurons communicate and transmit messages to and from various parts of the body within the peripheral nervous system, a concept known as neurotransmission. Classical neurotransmission features several key steps. These are the synthesis of the neurotransmitter, which is the molecule responsible for communication, mechanisms for its storage and release, as well as its removal from the synapse. Cholinergic neurons are neurons that release acetylcholine as a neurotransmitter. Acetylcholine is a small molecule with a positively charged quaternary ammonium group and a partially negatively charged ester group, which is rapidly hydrolyzed by acetylcholinesterase. Acetylcholine is the endogenous agonist for all subtypes of nicotinic and muscarinic receptors. Let's look at how acetylcholine fits the criteria of a classic neurotransmitter. Acetylcholine synthesis starts with the transport of choline into the presynaptic neuron. Acetylcholine is synthesized from choline and acetyl coenzyme A with the help of the enzyme choline acetyltransferase. It is then packed into vesicles via transporters and stored there. Once an action potential arrives at the nerve ending, the vesicles fuse with the terminal membrane and acetylcholine is released into the synaptic cleft, where it binds to nicotinic or muscarinic receptors on the postsynaptic neuron. Acetylcholine has a short half-life due to rapid hydrolyzation by the enzyme acetylcholinesterase, which breaks it down into choline and acetate to terminate its action. Choline is then recycled and acetylcholine is continually resynthesized and hydrolyzed. Due to its quick removal and non-selectivity for receptor subtypes, acetylcholine has limited therapeutic use, but other aspects of cholinergic neurotransmission remain valuable drug targets. Cholinergic neurotransmission serves many essential purposes within the peripheral nervous system. Another important neurotransmitter is noradrenaline. Adrenergic neurons make up the majority of postganglionic neurons of the sympathetic nervous system. The synthesis of noradrenaline starts with the transport of tyrosine into the presynaptic neuron. Tyrosine is converted into noradrenaline via a series of enzymatic reactions. First, tyrosine is converted to dopa by tyrosine hydroxylase, which is then converted to dopamine by dopa decarboxylase. Dopamine is subsequently converted to noradrenaline by dopamine beta hydroxylase. This occurs in vesicles at nerve endings, where noradrenaline is also stored until an action potential arrives. Similar to cholinergic neurotransmission, vesicular exocytosis is responsible for noradrenaline release into the synapse, where it binds to alpha and beta adrenergic receptors. However, unlike acetylcholine, the action of noradrenaline is terminated mainly through reuptake by noradrenaline transporters at the presynaptic nerve terminal. Due to longer half-lives and their functions as circulating hormones, both noradrenaline and adrenaline are therapeutically useful, and various aspects of their transmission also serve as drug targets. In summary, neurons communicate via neurotransmitters released from nerve terminals. The mechanism of neurotransmission involves neurotransmitter synthesis and storage in nerve terminals, its release into the synapse, and its removal or termination of action. Cholinergic and adrenergic neurotransmission governs neuronal communication in the autonomic and somatic nervous systems. Cholinergic and adrenergic neurons make up different parts of the peripheral nervous system.